Hello friends, once again we are here with our next video lecture based on fundamental unit of life. In last video conferencing lecture we have seen about what is meant by fundamental unit of life, why it is called as fundamental unit of life. The life starts from cell, so cell is called as a fundamental unit of life. In that we have seen who discovered cell, then who named cell, who has observed first living cell, then we have discussed about who has observed nucleus first, then after we have said about cell theory, Schleiden and Swan, then Virchow advanced cell theory. About cell theory we have discussed, then we have discussed about cytoplasm, we have discussed about protoplasm, we have discussed about osmosis, we have discussed about diffusion, we have discussed about prokaryotic cell and eukaryotic cell. We also have discussed about unicellular organisms and multicellular organisms. We discussed about differentiation of work or distribution of labor in the cell and in the organism. Then we have discussed about cell membrane, which is semi-permeable, only limited or selected material are allowed to pass through this membrane. That's why it is also called as a selectively permeable membrane. Then we are going towards next part of this chapter, that is different components of the cell. In last period we have seen about different components of the cell as cytoplasm, nucleus and cell membrane. So we have discussed about cell membrane is a bounding the material which is present inside the cell intact together, that is cell membrane, and that is a selectively permeable membrane. Only certain material are allowed to pass through it. Next part is cytoplasm. In cytoplasm, this cytoplasm was discovered by Kolliker in 1862. It is the site for both biosynthetic and catabolic pathways. Now what is meant by biosynthetic? The synthesis or preparations, and catabolic means breakdown. So breakdown pathways, it is also called as a metabolism, catabolism plus anabolism. Biosynthetic pathways are called as anabolic pathways, and the pathways which are breaking down the material to obtain energy, they are called as a catabolic pathways. So it can be divided into two parts. So cytoplasm, cytosol and cell organelles. So cytosol is nothing but the protoplasm we discussed. Aqueous soluble part contains various fibrous proteins forming a cytoskeleton, that is cytosol. And what is meant by protoplasm? Protoplasm is a liquid present inside the cell membrane apart from the cell organelles, that is called as a protoplasm. Then next part is cell organelles, living part of the cell having definite shape, structure and functions bounded by plasma membrane. So that is plasma membrane. Now what are the different parts of the cell or different organelles present inside the cell? Let's go from endoplasmic reticulum. So endoplasmic reticulum, what is its function and what exactly it is? This endoplasmic reticulum is made up of lamellae, cisternae and ribosomes. So endoplasmic reticulum, there are mainly of two types, smooth endoplasmic reticulum and rough endoplasmic reticulum. Endoplasmic reticulum, smooth endoplasmic reticulum and rough endoplasmic reticulum, SCR and RER. It is a network of membrane present in the cytoplasm. It is discovered by Porter, Cloud and Pullum. These are present in cells except prokaryotic and mammalian erythrocytes. Prokaryotic cells में एंडोप्लाज्मिक रेटिकुलम नहीं होगा, और mammalian erythrocytes यानी कि mammalian RBCs, erythrocytes means RBCs, ममालिया ग्रुप के ऑर्गेनिज्म के आरबीसीज में एंडोप्लाज्मिक रेटिकुलम नहीं होता है. रादर देन इन ऑल सेल्स this endoplasmic reticulum is present. They are made up of three components. They are called as cisternae, vesicle. Cisternae are, these are long flattened parallelly arranged unbranched tubules. These from successive layers of nucleus. These are found in cells which are active in protein synthesis and are 40 to 50 nanometer micrometer in diameter. These are called as cisternae. Vesicles, these are around or spherical. They are found in synthetically active cells. So there are two types of endoplasmic reticulum: smooth endoplasmic reticulum, rough endoplasmic reticulum. What is the difference between SER and RER? In smooth endoplasmic reticulum, they are made up of tubules mainly, whereas rough endoplasmic reticulum made up of cisternae only, cisternae and vesicles. This SER will help in steroid lipids and polysaccharides, storage of lipids and polysaccharide synthesis. Helps in RER, it helps in protein synthesis, contain ribosomes on its surface. So basic difference, or mainly if we want to categorize them really, then it is simple one, that is RER consists of ribosomes on them. SER doesn't have ribosomes on them. RER, that is rough endoplasmic reticulum, is present around the nucleus. Smooth endoplasmic reticulum is suspended in the cytoplasm. So this is the main difference. Small endoplasmic reticulum helps in membrane biogenesis. Whatever plasma membrane is there or nuclear membrane is there, all these membranes are biosynthesized. So these are synthesized with the help of smooth endoplasmic reticulum. What are the functions of endoplasmic reticulum? It is the only organelle which can move within a cell. So it serves as a channel for the transport. So it can be considered as a transport material. So there are two types of transport, public transport and goods transport. Like that, here one function is here, transport of material between various regions of cytoplasm and between cytoplasm and nucleus. For this it will work. One more transport system is present inside the cell, that is vacuoles. About them we will see in later these coming minutes. Now it is also function, endoplasmic reticulum also functions as cytoplasmic framework to provide space for some of the biochemical activities. It forms endoskeleton of cells. It helps in synthesis of fats and steroids, cholesterol, etc. It contains secretory proteins. Smooth endoplasmic reticulum play a crucial role in detoxification of drugs and poisonous byproducts inside the cell. Next cell organelle is Golgi apparatus. Golgi apparatus is made up of vacuoles, cisternal space, cisternae, sacules, cisternal membrane. All these are the parts of Golgi apparatus. This Golgi apparatus consists of system of membrane bound vesicles arranged parallel to each other in stacks called cisternae, along with some large and spherical vacuoles. It was discovered by Camillo Golgi, so that's why it is named as a Golgi apparatus. In plants, Golgi membrane bounded. Golgi apparatus is membrane bounded. It is single membrane bounded. It is absent in prokaryotes, mammalian RBCs and sieve cells. So what are the functions of this Golgi apparatus are? It helps in formation of lipids. It helps in formation of middle lamellae. It is a secretory in nature. It helps in melanin synthesis. Lipids and proteins synthesized in endoplasmic reticulum are packed at Golgi complex. They provide the site for assembly of new membrane material. So main function of membrane bound organelle, synthesis of membrane bound organelles is done by the Golgi apparatus. Now next part is mitochondria. Now what is meant by mitochondria? Mitochondria are the sites for synthesis of energy. Actually, these are the sites for breaking down the food material. We know that in the in photosynthesis, food is prepared. In the process of respiration, food is breakdown to release energy. In case of mitochondria, it helps to break down of energy. So energy is synthesized here. So mitochondria are also called as powerhouse of the cell. Mitochondria are also called as a powerhouse of the cell. Question may be asked in the examination that what is called as a powerhouse of the cell. So answer will be mitochondria. So these mitochondria help in the breakdown of the food material and they are called as a powerhouse of the cell. It is a rod shaped structure found in cytoplasm of all eukaryotic cells except mammalian RBCs. These are also absent in prokaryotes. So mitochondria are absent in prokaryotes because in prokaryotes membrane bound organelles are absent. So here it is absent in prokaryotes. It was first seen by Kolliker again in insect cells. Maximum mitochondria are found in metabolically active cells. It is also called as powerhouse of the cells. Just now I told you the storage battery. It is double membranous structure where outer membrane has specific proteins while inner membrane is folded inside to form chambers called cristae. Cristae are the enfoldings of inner mitochondrial membrane that possess enzymes of respiratory cycle like Krebs cycle. ATP synthesis units are also called oxysomes or F1 particles. Space between inner and outer mitochondrial membrane is called as a perimitochondrial space. The fluid present in mitochondria is called as matrix. So what are the functions of mitochondria? Its main function is to produce and store the energy in the form of ATP. It is the site of Krebs cycle of respiration. Oxysomes contains enzymes for ATP production. Matrix contains enzymes for Krebs cycle. These are the functions of mitochondria. Mitochondria are powerhouse of the cell. Just now you have to remember this, where breakdown of food particles takes place. Next cell organelle is ribosomes. So let us see the figure of ribosomes, how they look like or where they are present inside the cell. Ribosomes are the sites of protein synthesis. All structural and functional proteins, that is enzymes coded by the nuclear DNA, are synthesized upon cytoplasmic ribosomes. The DNA codes are transcripted to messenger RNA, that is mRNA molecules in the chromosomes of the nucleus. mRNA molecules diffuse out into the cytoplasm and each become attached to several ribosomes, which thus from a group called polyribosomes. In this way, each mRNA molecule brings about polymerization of specific protein molecules with the help of ribosomes from amino acid molecules found in the cytosol. So a little bit complicated. Let us just remember ribosomes are the sites for protein synthesis. Whatever proteins are synthesized in the cells, all these proteins are synthesized via ribosomes. Actually, the message which has came from the nucleus about synthesis of protein, because these proteins are nothing but the enzymes, and these enzymes will help in the all the metabolic activities, because without enzyme any reaction will not take place, no reaction will take place in our body. So synthesis of enzyme is required. Enzymes are nothing but the proteins, and these proteins are synthesized by ribosomes with the help of transcription, came from nucleus, came from DNA, which is genetic material, came from DNA via or with the help of mRNA. So ribosomes are called as a sites for protein synthesis. There are two types of ribosomes. They are 80S type of ribosomes and 70S type of ribosomes. 80S type of ribosomes are present in eukaryotic type of cells. 70S type of ribosomes are present in the prokaryotic type of cells. Yes, 70S means Sedberg unit. This is the centrifugation unit. 80S, 70S, these are the two types of ribosomes. Now after this, let us see about the next cell organelle, that is called as a plasters. Let us see the structure of plasters, how they looks. So these plastids are double membrane discoid structures found in only in the plant cells. Do remember, plastids found only in plant cells. They are absent in the animal cells. Plastids are of two types. They are leucoplast and chromoplast. Leucoplast and chromoplast. Leucoplast are colorless plastids, whereas chromoplast are color plastids. What plant have their own color? Leaves have their own color. This color is due to plastids. These plastids, in simple language, till now you people are known about plastids with the name chloroplast. So chloroplast is subtype of chromoplast. So besides being discoid or rhomboid in plants cells, they occur in variable shapes, like in algae they can be U-shaped, spiral, coiled, ribbon shaped, etc. depending upon type of pigments present in the plastids. I told you there are two types, but we can consider them as three types also. Leucoplast, no pigment system present. Chromoplast, color pigments, that is other than green, brown pigments, red pigments are present. Some plants leaves are red color, brown colored, so they are chromoplast. And chloroplast, green pigments, chlorophyll is found in them, so they are categorized into three. Now what is the main function of chloroplast? We know that synthesis of food material, that is photosynthesis. So where food preparation takes place, we call it as a kitchen. That's why plastids are called kitchen of the cell. Plastids are called as a kitchen of the cell. Like mitochondria are called as a powerhouse of the cell, plastids are called as a kitchen of the cell where synthesis of food takes place. Chloroplast have two parts, grana and stroma. So this grana consists of laminar system. So here collection or say harvesting of light takes place in grana, and stroma, granular transparent substance also called as a matrix. These grana are embedded in the stroma. Besides grana, they also contains lipid droplets, starch grains, ribosomes, etc. So what are the functions of chloroplast? This is the site for photosynthesis, also helps in protein synthesis due to the presence of ribosomes. So next part, let us see about vacuoles. These are membrane bound regions in the cytoplasm containing water and other substances. They are bound by single membrane called tonoplast. In animal cells, vacuoles are smaller in size and numerous, while in plant cells, single large vacuole is present. So if you want to distinguish between plant cells and animal cells, if you want to identify whether which is plant cell or which is animal cell, simple technique is there. Cell wall is there. First of all, plant cells, in plant cells cell wall will be present. In animal cells there will be no cell wall, and in animal cells many vacuoles are present, whereas in plant cells only one vacuole, large vacuole is present. In animal cells small vacuoles are there, in plant cells there are large vacuoles. So what are the functions of vacuoles? I told you in endoplasmic reticulum we have seen is a transportation system. Likely vacuoles also have a function of transportation system. Also along with it helps in maintaining osmotic pressure in cell. I told you plant cells cell wall is present. Suppose there is a scarcity of water and cell have to maintain its turgidity, it maintain its strength, inner whatever pressure is there, it should be maintained. So in that case vacuoles will become larger and they will maintain more osmotic pressure in the cell. It stores toxic metabolic products of the plant cell. It contains various color pigments like anthocyanin. So about different pigments, chlorophyll pigments, anthocyanin also is a pigment. It gives color. Lysosomes is the next cell organelle which we have to discuss about. So these lysosomes were discussed about Kristen A. Dewey. Lyso means digestive, some means body, digestive bodies means lysosomes. So we have seen that mitochondria are called as a powerhouse of the cell, chloroplast or say plastids are called as a kitchen of the cells, and lysosomes are called as suicide bags of the cell. They are called as a suicide bags of the cell. These are tiny sacs like granules containing enzymes of intracellular digestion. They are bound by a single membrane. They occur in animal cells and a few plant cells. They do not have a definite shape or size. They contain hydrolyzing enzymes called acid hydrolysase. Mainly concentrated acids are present inside the lysosomes. Whatever debris material, waste material is present in the cells, all that waste material is sent to the lysosomes, and due to concentrated acids present in the lysosomes, lysosome digests that material, destroy that material. But suppose if cell undergo at some problematic case, or that cell is not performing well, at that time this bag of lysosome get burst, which contains concentrated acids, and it spreads in the cytoplasm and it kills cell. That's why it is also called as a suicide bag of the cell. These lysosomes helps in digesting foreign material and ohm out cells. They are kind of waste disposal system. That's why these lysosomes are very important in the cells, because waste material need to be get disposed out. For that these lysosomes are helping. Next part is peroxisomes. These structures were first described from liver and kidney by Rodin, this is the name of scientist, in 1954. In plant cells they were first observed in germinating seeds by Tolbert. The term peroxisomes was first used by Dewey and also called eurycosomes. Peroxisomes are ovoid or granular structures limited by a single unit membrane and have a diameter of 0.5 to 0.9 micrometer. In green leaves of C3 plants, peroxisomes carry out photorespiration. So what is the function of peroxisomes? Photorespiration. During photosynthesis, again there is a requirement of respiration, so that requirement of respiration is fulfilled by peroxisomes. In animal cells they are carry out lipid metabolism. They contain important enzymes as oxidase, peroxide producing enzymes, peroxidase and catalase, which break down toxic peroxides to water and oxygen. So this is about peroxisomes. Then glyoxisomes are different organelles, cell organelles present in the cell. A Beaver, that is 1961, was the first person to discover these organelles and were described later by Bredbach. They are about 0.5 to 1 micrometer in size and are surrounded by a single unit membrane. They are found in plant cells, particularly in germinating fatty acids seeds. Glycosomes contains important enzymes, and the structure of glycosomes is similar to peroxisomes. So now let us see the difference between plant cell and animal cell. In plant cells, cell wall is present. In animal cells, cell wall is absent. Plastids are found in plant cells. Plastids are absent in animal cells. One large vacuole is present in plant cells. Numerous tiny vacuoles are found in animal cells. In plant cells, centriole is absent. In animal cells, centriole is present. On the basis of type of organization, cells can be differentiated into two types: prokaryotic cells and eukaryotic cells. What are their differences? Let us see. In first period we have already discussed. In prokaryotic cell, single closed loop is present, that is histone is absent as far as DNA is present, DNA is concerned. In eukaryotic cell, multiple chromosomes are present. So chromosomes in chromosomes DNA are packed. In prokaryotic cells, respiratory system is in plasma membrane. In eukaryotic cell, it is with the help of mitochondria. Photosynthetic apparatus in prokaryotic cell is internal membrane, and here in eukaryotic cell it is chloroplast. Golgi body is chloroplast, endoplasmic reticulum, mitochondria, lysosomes, all are absent in prokaryotic cells, whereas Golgi bodies, chloroplast, endoplasmic reticulum, mitochondria, lysosomes are present in the eukaryotic cell. Ribosomes, 70S type of ribosomes are present in prokaryotic type of cells, I just told you, and 80S type of ribosomes are present in the eukaryotic cell. Cell wall generally present in prokaryotic cells. It is complex chemical composition. In eukaryotic cells, it is present only in plant cells. Eukaryotic cells present in some types. Simple chemical composition is there. Flagella, prokaryotic cells properly, and in some in eukaryotic cells, flagella are present in some cases. Cytoplasmic streaming rare or it is also absent, and in case of eukaryotic cells, cytoplasmic streaming often occurs. Vacuoles are absent in prokaryotic cells, vacuoles are present in eukaryotic cells. Lysosomes are absent in prokaryotic cells, lysosomes are present in eukaryotic cells. Capsules may be present in prokaryotic cells, they are always absent in eukaryotic cells. Hereditary motility is DNA in circular form, and in eukaryotic cells DNA is linear. So apart from this, we have to just see about cell division. So how cells will be get divided? Because no cell is eternal, they have to divide, they have to change from one life cycle to another life cycle, isn't it? So how cells will be dividing? There are two types of cell division, that is first is mitosis and second is meiosis. In case of mitosis, cell divides with same chromosome number. Cell divides with same chromosome number, whereas in meiosis chromosome number reduced to half. In case of meiosis, chromosome number reduced to half. Then in case of mitosis, from one cell there will be formation of two cells. One cell get divided into two cells and chromosome number maintains same. In case of meiosis, one cell get divided into four cells where chromosome reduced to half, chromosome number reduced to half. Mitotic cell division takes place at somatic cells. Mitotic cell division takes place at somatic cells, means body cells, apart from gamete cells it takes place, whereas meiosis takes place at gametic cells, where formation of sperms or formation of egg cells, formation of golden grains takes place. At that point meiosis takes place, whereas in our whole body, cheek cells, nose, head, legs, hand, wherever dead cells formation takes place, that at that places mitosis takes place, which maintain its chromosome number. Why in meiosis chromosome number reduced to half? Because at the time of reproduction these are gametes which are chromosome number are reduced to half, that is gametes, which further goes and fuses to form zygote. So 50% chromosomes from parental side, either parental side, that is from mother, and 50% chromosome from father, they come together to once again form equal chromosome number which is present in the zygote, and so chromosome number maintain same generation after generation. So this is the beauty of meiosis, which maintain chromosome number same as it is in sexually reproducing organisms. So in case of mitosis, again this mitosis helps in maintaining the cell structure, body cells as it is. So students, we have finished here our first chapter, and whatever DPPs are given or exercises are given in the book, you have to complete it and you have to report it to me as soon as it get completed. Stay home, stay safe. Thank you very much.